Swayam Prabha. Digital India. Educated India.
Students, welcome back to the Developmental Biology course. In the last class, we were discussing the very early part of the mammalian embryonic development up to the end of blastocyst, and then we looked at the various names like epiblast, embryonic epiblast, amniotic ectoderm, hypoblast, and so on. So today we are going to continue that from gastrulation onwards. So one important thing that we discussed in the last class is the molecular events that determine who is going to become trophoblast and who is going to become the inner cell mass. Okay, so this inner cell mass and the trophoblast are the key initial differentiation that happens in the very early stage of the embryonic development. So today we continue from there. This is another view of whatever we have seen. So yesterday we saw a cartoon like this. Yeah, here we have the last point. So we saw something like this. The same thing, a different view, a sort of a 3D view is what you have in this. So this is the embryonic epiblast. After having epiblast given rise to this amniotic ectoderm, this blue color here, and this is the amniotic cavity, this is the yolk sac. So this is the extra embryonic mesoderm that came from the hypoblast. Okay, so this green cells. So these two layer of cells here, if you can imagine a 3D structure, it's going to be like a disc-like thing. Okay, so here this is another view of this, where you are seeing the surface of this area, the grayish area. Okay, so this is the embryonic epiblast, and the greenish thing below that is equivalent of this. So these two discs together are called the bilaminar germ disc, okay, from which everything comes, primarily actually from the epiblast. And so this is our starting point of gastrulation. Okay, so the gastrulation gets over by the 15th day in human after gestation. So the main events of gastrulation is basically these cells start migrating in between the two layers as shown here. So the formation of this what we call as the primitive streak. So this is the posterior and this is the anterior, and this is the node. Okay, so this node and primitive streak are structures through which the cells are going to go like this, go inward between these two layers. Okay, this gray and the green layers between the two, and that is how we are going to have the endoderm and mesoderm going to form. So that's the key event, endoderm and mesoderm form from epiblast. And the migration direction, so these arrows are darkened just to highlight the point that they are beneath this cell, the gray cell layer, okay, between these two. And this white arrow says from the top where it is going to go like this. And so if you take a cross section along these dotted lines and look at them, so you are going to see like this. So this is a 14th to 15th day, and this is the 16th day, okay, at the end of 16th day. So here what you see is from the epiblast cells you get these endoderm cells, these yellow colored ones coming down, which is going to be later followed by mesodermal cells. So these cells are actually epiblast cells, and some of those cells lose E category in expression, and therefore adhesion is lost and they become migrating epiblasts. and these are detached from the neighboring layers of cells and then they migrate down. Okay, an important signaling pathway that regulates is the FGF. So FGFs coordinate specification of these two types of cells, that is endoderm, mesoderm, as well as they help their migration during gastrulation. And one of the potential downstream targets of FGF is the E-cadherin. So E-cadherin level is probably downregulated by the FGF signaling, and therefore that facilitates the detachment of these cells. And also it regulates the expression of these transcription factors, SNIL, BRCA and TBX6, and these are essential for those fate specifications. So this is the main point of gastrulation. So much of these processes, so eventually this endoderm that is coming out is going to replace these hypoblast cells, and that's going to form layer here. So much of our understanding of mammalian embryonic development actually comes from studying the mouse embryo development, and mouse embryo at this stage is not flat disc like this. Instead, this disc starting at the node is folded up. Okay, so this gray sheet is folded up along with the green below, and therefore this endoderm here is going to be on the outer side of the embryo. So that we will see in the next slide. Okay, but before we go further to that step, we need to look at what is happening outside of the embryo. So far I was talking about these two cells, and we shouldn't forget about these trophoblast cells. What are they doing simultaneously while the embryonic gastrulation is happening? So they are busy setting up the connection with the mother, okay, and that is explained in this. So if you recall from the last class, the outer layer, the trophoblasts, they form a layer which we call syncytio trophoblast, and they produce the proteases that help in dissolving ECM and getting attached to the endometrium, and then they give rise to a set of cells where they undergo nuclear division without cytokinesis, forming multinucleated cells called the syncytio trophoblasts, like these cells, this light pink. And these are the ones that end up setting up the actual connection. So they form the chorion of the placenta. Okay, so this detail we need to understand clearly, and that's the focus of this slide. So the trophoblast cells give rise to cytotrophoblasts, and these are the ones that are important for attaching to the endometrium initially, and they accomplish this by producing proteolytic enzymes, and that enable them to enter into the uterine wall and remodel the uterine blood vessels. And this job is taken up to a further extent by the syncytio trophoblasts. So these are called syncytio primarily because these are multinucleate, so therefore syncytium. Okay, so syncytial trophoblast is syncytio trophoblast. Further the progression into the uterine wall. So eventually what they are going to do is they are going to line the maternal vascular tissue. So basically they are going to dissolve. See, this is the in the bottom image. So we have a close-up view in the next slide. Yeah. So this is the blood vessel from the mother, so the mother's tissue, and these trophoblast cells are going to line those blood vessels. See, you see here the blood vessel basically now ends here and it gets lined. It opens up into a wider area rather than being a tube-like structure. It opens in a wider area lined by these trophoblast cells, syncytio trophoblast cells. Okay, and the extra embryonic mesoderm that we saw earlier, like these cells here, this coming up here, a couple of them are visible in this and one in this side. So these are the ones that are eventually going to form the blood vessels from the fetus side, okay, and that is what is going to give rise to the blood vessels within the umbilical cord. Okay, so umbilical cord, so as you see here, is the connection between the fetus and the mother, and within that, so that umbilical cord itself and these blood vessels inside this are formed by these extra embryonic mesoderm. Okay, so they form these blood vessels, and these actually differentiate into these loop like structures which we call as the chorionic villus, and that increases the surface area a lot. And this is quite clear if you see in this actual photograph of an embryo. So this is the amniotic sac, inside with you see the embryo, and this is the trophoblast cover, and this is the chorionic villi. Okay, so this portion that you see, so that's here, formed by the extra embryonic mesoderm, and so that's what forms the chorion. So this portion is chorion, so along with this trophoblast cell layer as well, and the mother's blood bathes this area. So these tubes, the blood vessels of the mother and the fetus, are not connected, they are not continuous tube like structures. Instead they exchange in this area in which both of them diffuse. So the oxygen and other food supply from the mother's circulation comes into this area from which this chorionic villus takes up. And similarly, urea and carbon dioxide from this diffuses into this area from which it drains into the mother's vein. Okay, so this is how the placental structure actually forms. So this portion, which is modified by the endometrium, is the decidua and mother's part, and this portion coming from the trophoblast derived cells and extra embryonic mesoderm forms the chorion, and that is the fetal component of the placenta. Okay, and this connecting thing is what we call as the umbilical cord. So I hope you have a clearer idea of what is chorion and what is decidua and what actually is placenta is. One point I want to highlight is there is no direct blood vessel connection here. So they both bathe a common area which allows the diffusion between the two circulatory systems. So this gives you a closer view of the structure that we just discussed. So you have the maternal artery flowing into this area, and then the maternal vein that drains from this area, and you need to focus on this. So you actually have umbilical cord artery, you have two of them, while the vein there is only one. Okay, so we now step back a little bit and start from inner cell mass once again to understand one specific aspect of inner cell mass. So as we discussed several lectures ago, while talking about specification, different kinds of specification, I mentioned that mammalian, actually all vertebrates follow what is called regulative specification leading to conditional specification leading to regulative development. Okay, so where interaction among the neighboring cells are critical. Okay, so no cell comes specified by the maternal inheritance. Okay, so therefore if you remove any one cell in the inner cell mass, it doesn't matter, the remaining cells will replace its part. So each inner cell mass is actually capable of forming an entire embryo. So they are completely undifferentiated, except that they don't make trophoblast. They are capable, each one of the inner cell mass cells are capable of making the entire embryo. So there are certain implications of this property, and that is highlighted in this slide and in the next slide as well. So for example, if you start with 2-cell embryo, and let us say when they form the they reach the blastocyst stage, so inner cell mass cells are in one end of this blastocele, so this is blastocyst, okay, it is no longer blastula. And suppose, let us say, it split into 2 groups before this step itself, okay, so between 2-cell stage and the morula stage, if it separated into 2, and they are capable of forming then 2 embryos. Okay, those cells are capable of making both the trophoblast, inner cell mass, and as a result you end up if it is split into 2 parts, if the embryo is split into 2 parts that early, like for example in 8-cell stage or earlier, before the morula stage, any stage, if it splits into 2, then they end up making 2 trophoblasts and 2 inner cell mass, leading to making 2 chorions, because chorions come from trophoblast, and then the inner cell mass giving rise to epiblast, from which you get the amnion, and as a result you get 2 amniotic cavity as well. So 2 chorion, 2 amnion, and you get this sort of zygotic twins. Okay, so these are not fraternal twins. Fraternal twins arise from 2 oocytes getting fertilized by 2 sperm at the same time and developing. In one pregnancy you have 2 fertilization events, that is fraternal twins. So we are not talking about that here. We are talking about twins that arise from single fertilization, and this is possible simply because of the totipotency of early embryonic cells. Okay. Now suppose, let us say, the split happens later after the trophoblast formed and the inner cell mass split into 2 groups, and now they are going to make 2 epiblasts and hypoblasts, and as a result that epiblast is going to give rise to each of them going to give rise to 2 amniotic ectoderm, and therefore you will end up making 2 amniotic cavity. But this happened after the trophoblasts separated, and due to that you are going to have a single chorion. So single trophoblast population, and as a result single chorion, but 2 inner cell mass, and as a result you get 2 amniotic cavity. So here you can see this shaded area, that is a trophoblast, and that is the chorion there formed from the trophoblast, and this white area is the amniotic cavity. So you have 2 amnion with 1 chorion. And suppose if it happens even later, like for example the embryonic epiblast separate into 2 groups after the amniotic ectoderm formed, and therefore a single amniotic cavity, but the embryonic epiblast split into 2. Now you get 2 embryos in a single amnion and single chorion. So these sort of twinning during embryonic development unequivocally establishes the pluripotency of the inner cell mass cells. At all these stages the cells are capable of forming an entire embryo. So if splitting is possible, then mixing also should be possible, right? And that is what is illustrated in the next slide. So here you have 2 different pigmentation, like the fur color as you see here, 2 different fur color. Okay. So if you take embryos from the 2 different fur colored mice and open up the zona pellucida and allow the the cells to mix, for example, it is readily doable before compaction, right? And if you take the cells and mix them, and they form its regulative development, so therefore they form a single inner cell mass and form an embryo that develops into an adult mouse with mosaic color pattern. See, as you can see here, the cells of both genotypes are interspersed and resulting finally in this mosaic color pattern. Okay. And you can take this further. Suppose let us say this mouse mates with a homozygous recessive, that is the gene for which this fur color is encoded is recessive and does not produce any pigments, and that is the white. Okay, white is the homozygous recessive phenotype. And let us see what happens. So, in this experiment actually 3 different pigmented morullas were taken and fused into one, and they form 3 different colors. You see white and this brown and then the black, all 3 mosaics. And these cells contributed to the germline as well. Okay, the germ cells have been derived from these cells, and as a result you have 3 different genotypes of germ cells, and when oocytes formed from those 3 different types developing in a single mother, when they are fertilized by sperm from a homozygous recessive, then you end up getting all 3 colors separate in the progeny. So, the black gene, where in whichever oocyte that was present, that when fused with a sperm coming from with the white allele, then that gives rise to black. And if the germ cell had the brown allele, this homogeneous brown fur, and if it had the recessive allele, and then with the recessive allele from the sperm, then you have homozygous recessive and therefore a homogeneous white fur. So, both of this very clearly show the potential, the pluripotency of the inner cell mass to embryonic epiplast stage. So, now let us get back to the development of the embryo. So, I told you at the end of gastrulation, so I told you that this disc, if it is folded upward with this node at the bottom, so how would that look like? In that, this will be inside and this will be outside, and that is the case in mouse. So, we get to that now and then continue further with the embryonic development. Yeah. So, this is the cup shaped embryo. So, this is the node, and the posterior side, the primitive streak is here, and this is the anterior part. Okay. So, the endoderm is outside. So, this AVE, anterior visceral endoderm, that actually formed from the hypoblast initially, and that is at this anterior most end. Posterior is often called caudal, spelling for that is here in a second. Okay, this is often used in developmental biology, and it just means posterior. So, this is the caudal side, and this is the anterior side where the head is going to form, and the node is right in the middle at the bottom. So, this is the primitive streak. So, between this blue and then sort of greenish grey area, so between these two you have the migrations of the cells happening as shown here. Okay, so this is a later stage. So, so now our focus is, okay, the gastrulation we sort of understood, the endoderm and mesoderm start to migrate through the primitive streak and the node, and now how the head to tail axis is formed. So, that is the focus of the next few slides. So there are two signaling centers basically. One is formed by this anterior visceral endoderm at the near the head area where the head is going to form, and then at the bottom of this cup shape you have the node. So that is the one from where the cells are going to fold and form a tube-like structure that will finally connect from here to the head, and that's going to be the notochord. And this node and this AVE form the two signaling centers that determine the head to tail axis. So we haven't understood this process in as much detail as we saw in Drosophila anterior posterior axis formation, but some basic details are emerging already. For example, this AVE produces lefty and cerebrous proteins, and these produced here antagonize nodal expressed in the epiblast cells, and therefore nodal is not going to be expressed in this area, and that seems to be key for positioning the node and primitive streak in this part and not here. And so that is one key aspect, that is production of lefty and cerebrous by AVE, and that suppresses the nodal to the posterior part. And later on, when the node starts producing chordin, and then when notochord is produced from the node area, when the cells form the notochord, and this node formation itself is induced by the underlying trophoblast cells, and they seem to be key in specifying the node. And the node itself is derived from the mesodermal layer, and the cells coming from that are these precaudal mesoderm in this area here. Okay, so this is coming from the notochord, and this once node produces the notochord produces noggin, and this chordin and noggin are key along with this AVE for the head structures to form. And what they do, they actually are antagonists of the BMP signaling. So BMP gets restricted to the posterior, and so chordin and noggin are important for the head structures to form, okay, including the neural tube. So this is illustrated by the phenotypes of the chordin and noggin knockout mice shown here. So this shows this is a whole embryo stained for chordin expression. So chordin is there in the anterior part of the primitive streak. See, this will be the primitive streak. So this is where the node is going to form, and then it is there in the node and in the anterior structures here. Okay, so that's what you are seeing as the embryonic development continues. So that's the chordin expression pattern. And if chordin is knocked out, except this ear development defect, so this is the knockout and this is the wild type, the head structure formation appears to be totally alright. But if chordin and noggin both are knocked out, the head structure doesn't develop right. There is no jaw formed and only one eye, and that is below a nose-like protrusion, but otherwise no clear head structure forms. So indicating that these probably function in a redundant fashion required for the head structure to form. And another important molecule that plays a role in this head to tail axis formation is the vitamin retinoic acid, vitamin derived signaling molecule retinoic acid. This is from vitamin A. Okay, so retinoic acid can alter this anterior posterior axis if it is introduced at the stage of gastrulation. So when embryo is incubated with retinoic acid, so you have alterations, which is illustrated here. So TBX is a transcription factor normally expressed in the anterior primitive streak. So this is the primitive streak and this is the anterior part of it and the surrounding epiblast cells. So this is where TBS6 is normally expressed. And when embryo is bathed in retinoic acid that alters this anterior posterior axis formation such that it's duplicated into two axes, one here and one here. Okay, and then you have two this kind of TBX expression pattern in this embryo. And another critical signaling molecule, the sonic hedgehog, is expressed strongly in the notochord here. And now with retinoic acid application, it gets split into two notochords. Here you can clearly see two structures. And this is probably one of the causes for the formation of conjoined twins. In TVs you would have seen the images of some children, even grown up adults, where they are joined at the head. So that is due to head to tail axis, partial duplication of that axis, and that is what leads to the formation of conjoined twins. Alright, so this sort of summarizes what we discussed as well as introduces some more details. So this is flattened disc of the embryo, this cartoon representation. This is the anterior and that's the posterior most at the end of gas stage. And we didn't talk about organizers. Organizers probably in a later lecture I will introduce this if we are going to discuss amphibian embryonic development. And starting from the organizer, the anterior structures produce antagonists of these signaling molecules, Wnt, BMP and FGF. So FGF we already saw, and then I also told you the chordin and noggin are antagonists of BMP. So as a result, these molecules are concentrated in the posterior. So FGF mRNA transcribed here gets degraded as it moves anteriorly and forming an posterior to anterior gradient. And the antagonists form the opposite gradient because they are produced by organizer and the anterior structures. So you have two gradients, this FGF, BMP, Wnt forming a posterior to anterior gradient, and then their antagonists in the opposite. And so these molecules specify the posterior. So this is the tail bud region of mouse embryo where in situ hybridization shows the level of FGF8 mRNA, red being the strongest and green low. Towards the tip you have the highest concentration of FGF8 mRNA, and it progressively decreases as you go towards the anterior part. Okay. And so the retinoic acid forms another gradient. Retinoic acid producing enzymes are in the posterior, while retinoic acid degrading enzymes are in the anterior. So retinoic acid, in addition to these three signaling proteins, retinoic acid also forms a posterior to anterior gradient. So signaling from these molecules as well as retinoic acid all converge in the activation of the CDX proteins, which by acting on Hox gene coordinate all these signals into determining the anterior posterior patterning. So therefore now we move on to Hox genes. Okay, so the initial thing is you have two signaling centers, one near the head region, future head where it's going to form, and the other one, that is the AVE, and the other one is the node. And the AVE produces signals that restrict the posterior signals to the posterior and enable the head formation by the activity of chordin and noggin at a later stage. And that ends up setting up these two gradients that I just said, and in addition we have a retinoic acid gradient as well. So all of these gradients converge on Hox gene expression, and which Hox gene is expressed in which part along this anterior posterior axis provides the identity for those specific areas, what kind of organs are going to form there. So that is our next goal. Alright, so let us revisit Hox gene. I am saying revisit because we have already learnt about Hox genes when we discussed Drosophila anterior posterior axis formation. So we saw that Hox genes in the Drosophila are all on present on one chromosome in two clusters, the anterior cluster and antennapedia cluster, and the posterior cluster, which is the bithorax cluster. Okay, and I told you that they are arranged on the chromosome from the 5' end to 3' end, and they keep the same sequence, that is collinearity, in the way organs form from anterior to posterior. That is, Hox genes present on near the 5' most part of the chromosome specifies the anterior most structures in the anterior posterior structure axis, sorry, and similarly the posterior most structures are specified by Hox genes that are located 3' most on the chromosome. So that is the collinearity of their presence in the chromosome and their action along the anterior posterior axis. So initially their expression is in that pattern. The 5' most ones are expressed in the anterior most, and the 3' ones expressed in the posterior most part of the embryo, leading to those structure specifications in that order. So remarkably, that sort of a collinearity is conserved even in vertebrates, in all vertebrates. Okay, except that vertebrates have 4 copies of each of this. So this cluster, there is only one cluster in Drosophila corresponding to these body parts, and the very extreme anterior head region is determined by 2 separate transcription factors. Okay, so I will explain that a little later. So they are not specified by the Hox gene. So these uncolored, the grey region. So these are the ones that are color coded correspondingly to their locus, to their loci on the chromosome. Okay, so the 5' end anterior most and 3' one to the posterior most in the anterior posterior axis. Okay, and this is exactly the same order you see in the vertebrate chromosomes, except that there are 4 copies on 4 different chromosomes. For example, in mouse you have Hox A set in one chromosome, Hox B set in another one, Hox C in third, and Hox D in the fourth one. So this A1, B1, D1, etc. are paralogs. The same sequence present in multiple copies on the same organism are called the paralogs. Okay, so these are sequence wise most closely related to each other than to the other Hox genes, and so on. Like for example, if you take A9, so A9 is an ortholog of the Drosophila abdomen B, and that has 4 copies, meaning 4 paralogs. So abdomen B is ortholog, meaning similar sequence in another organism, and similar sequence in the same organism you have 4 paralogs. For some of them you have 4, some you have 3, and for some you have only 2. Okay, so it depends on the duplication events and the loss of the duplicates during the course of evolution. So for some of them you have 4 and some of them you have only 2 and so on. Okay. Yeah, I don't see any only 1 copy. So for every one of them you have at least 2. And here again the order is maintained. So as shown by the color codes here. Okay, in the dorsal part of the anterior posterior axis where you have the neural tube as well as the somites that form from the mesoderm as shown here. Okay, so now we move on to understanding a concept called the Hawkes code hypothesis. So it's extremely simple. What Hawkes code hypothesis says is, along this axis, head to tail axis, what structure is going to form where. So here we are only seeing the vertebral column. So we are looking only the dorsal part of the, that is top of the anterior posterior axis, top of the embryo along the anterior posterior axis is what we are looking at. So the different vertebrae here, like for example thoracic vertebrae has this, it forms the rib bones, that forms the chamber to protect the heart and lungs, and that differs in morphology. So this is a little longer than this. So this is like the neck and so on, okay, lumbar and sacral. So the which one is going to be what is determined by the combination of Hawkes genes expressed in that place, okay. The combination of Hawkes genes expressed in a given position along the anterior posterior axis specifies what structures are going to form there. They provide the segmental identity for a given position along this axis. That is what Hawkes code hypothesis states, okay. So is this true? It is true, and evidence comes from 3 different kinds of independent observations. One of them is comparative anatomy. So you look at the skeletal structure. For example, here you look at mouse skeleton. So its neck is lot longer than mouse if you look at this. So here are the rib cage, okay, and here this is the rib cage. So between the rib cage and the head, its long neck, and this is a short neck. And now when we look at the Hawkes gene expression pattern, quite interestingly, Hawkes 6 expression here extends more anteriorly, while the Hawkes 5 expression in the chick, so this is the bottom row is the mouse and the top row is the chick, and this extends posteriorly. And as a result of this, you end up getting longer, the Hawkes 6 is restricted to up to this, and therefore thoracic forms only up to that, and then you have a long region of cervical vertebrae. So you need to remember here the total length remains the same, the number of vertebrae are the same. It is just that either you have a situation where the cervical vertebrae are converted into thoracic, or the other way, depending on which one is your starting reference, whether it's mouse or chick. So this kind of supports the idea that which Hawkes gene is expressed determines what kind of vertebra is going to form, okay. So another important point I want to say by retracting a little bit back is I told you about this extreme head region, and they are determined by orthodentical and empty spiracles gene in the Drosophila, and believe it or not, their orthologs are the ones that determine the midbrain and forebrain, okay. So the Hawkes gene expression extends only up to the anterior most of the hindbrain. So the head region in organisms separated as much as 500 million years is determined by very similar genes. Remarkable indeed. So now another important point I wanted to mention before going ahead is a given segment's identity seems to be determined by the posterior most gene that is expressed here. For example, if you take here the posterior most gene expressed here is Hawkes 6, and that is what determines whether it is going to become thoracic or cervical. And this results primarily because the anterior gene's expression is restricted by the posterior gene expression. So in other words, if I remove Hawkes 6, Hawkes 5 will extend, okay. So that's what happens when you delete a Hawkes gene. the anterior structures shift posteriorly. And this is exactly what we saw when we were discussing about Antennapedia in Drosophila. Okay. So this is another important point we need to remember. So the posterior most gene expressed in a given position in the anterior posterior axis determines what segmental identity that portion is going to get. So this is comparative anatomy. The expression pattern, what Hox gene is expressed, seem to correspond to what kind of vertebra form in these two species. And the next we are going to look at gene knockout experiments. Okay. So here for example this is the wild type where you have this is the thoracic vertebra forming the rib cage, and then this is the lumbar between the your chest and the hip region. So that portion, in the ventral part you have your stomach. So that portion of the vertebral column looks like this. So now if you delete, so remember in mouse it's not like one ultra bithorax that you need to delete or one Antennapedia you need to delete. Here you have 4 copies, and therefore all 4 needs to be knocked out to get a phenotype. Otherwise they are redundant among themselves and they will lead to normal development. So when the 10th Hox 10, it's A, C, D, D, all are deleted, the 2 alleles of A, 2 alleles of C, 2 alleles of D. To highlight this further, here you have A, B, C, and if we are talking about a 10, you have A10 and we are diploid, mouse is also diploid, so you will have A10, 2 of them, 2 of this, 2 of this. So when you delete all of them, that is what is this. Then you have the expression of the 9 extending posteriorly, ending up producing rib cage like vertebra here in the lumbar region. Okay. Again supporting the Hox code hypothesis. The combination of Hox genes specify the segmental identity for in any given position along this axis. And in a similar experiment when Hox 11 is all the paralogs are deleted, you have the sacral region getting converted into lumbar like structures. Okay, so sacral forms this little extension and lumbar doesn't normally, and that's what now sacral also forms. So this is second line of evidence that supports Hox gene code. So the third is retinoic acid. So remember retinoic acid will mess up the anterior posterior axis, and we just saw that retinoic acid is expressed highest at the posterior where its synthesizing enzymes are present, and it is degraded as you move anteriorly. And then we also saw that retinoic acid via CDX genes impact on the Hox gene expression. So as shown in this cartoon. Okay. So strong retinoic acid higher concentration of retinoic acid means most posterior gene being expressed. So in an anterior portion, if you increase the retinoic acid, then you are going to have posterior Hox genes expressed there. And then we should also have the segmental identity shifting, the posterior structures shifting anteriorly, and that is what is seen in this. So this is the wild type, so thoracic, cervical, so that's the neck region, okay, this one. So you see that shift here. When retinoic acid concentration is increased by incubating the embryo at the appropriate stage in high concentration of retinoic acid, then you have additional the shift happening anteriorly. So as a result, as cervical vertebra gets converted into thoracic, forming these ribs here. Okay. So C6 becomes C7 and so on. And on the other hand, if you deplete the retinoic acid, then you have the opposite trend. Okay. C1, C2 becomes C1, and similarly here at this junction you will see T1 becoming C6, and therefore it is cervical and doesn't form the ribs. Okay. And this is also seen when you mess up with the Hox gene expression. So here Hox gene overexpression shifts anteriorly, and Hox gene mutations shift posteriorly, because I told you the posterior Hox genes try to suppress the expression of the adjacent anterior Hox genes. So as a result, when you increase a posterior gene expression, then it shifts anteriorly because it suppresses the anterior gene expression, and therefore the posterior expression domain shifts anteriorly, leading to posterior structures developing anteriorly. That's how this becomes converted into additional rib forming thoracic vertebral column, and the opposite with the loss. So these three evidences, one is the comparative anatomy, what is the morphology of the different vertebra along the anterior posterior axis in one organism, and another organism where the morphology is slightly different, and how are the gene expressions correlated with this. So that is the comparative anatomy that we saw here between chick and mouse, and this long neck corresponds to shorter expression of this HOX6 and this longer expression of HOX5 in chick when you compare with the mouse. And the second one is when you knock out all orthologs or paralogs of given Hox genes, we see the structures shifting posteriorly. And the third evidence is the increasing retinoic acid or decreasing retinoic acid. Similarly, Hox gene overexpression or underexpression. So these three lines of evidence support the Hox code hypothesis. Okay, and what is really remarkable is this pattern of Hox gene expression along the anterior posterior axis and the specification of segmental identity is remarkably well conserved almost all the way starting from flatworms up to us, that is Homo sapiens. Okay. So today I will stop here and we will continue to a new chapter where we are going to look at the evolutionary implications of developmental mechanisms in what we call as the evolutionary developmental biology. Okay. So that will be our next topic.